Hi everyone, my name is Gabriella Jessica, and today I'm going to talk about my project supervised by Mary Bird with the title Comparative Analysis of NetM gene function in simple leaf of Arabidopsis thaliana and compound leaf of Cardamine hirsuta. So a little bit of a background. The leaf itself is an important organ in the plant, serving as a site for important processes such as photosynthesis. In different plant species, there are different leaf shape, ranging from simple leaf, like on the left side here, a simple undivided leaf, like in Arabidopsis, to a more complex leaf on the right side, like cardamine and tomato, highly compound, highly lobes, and more divided leaflets. This knowledge in leaf morphology has been used as an application in agriculture. For example, this previous study used the genetic modification of leaf angle in rice. And as a result, as you can see here on the right side, the mutant has improved light interception in the canopy and resulted in higher grain yield. The tail homeodomain proteins are transcription factors that has a role in affecting leaf morphology and complexity. In a study, the overexpression of NOX1 gene, which is a part of the tail homeodomain proteins, changes the way the leaf shape, and it is done in two different plant species that have two different leaf morphology. In the figure A over here, the overexpression of NOX1 gene in simple leaf species create a more serrated leaf and smaller leaf, while the overexpression of NOX1 in compound leaf species create a more complex leaf with more division. This suggests that the tail homeodomain proteins has a role across different plant species and affect the leaf morphology and also the complexity. There are two classes of tail found in plants. The first one is knotted one like homeobox or knox. The second one is bell one like or bell. And NetM is a novel knox gene that falls into the third class of knox gene family. It encodes the main knox domain but lacks the homeodomain. Previous study of NATM in Arabidopsis showed that the overexpression and the ectopic expression of NATM affect leaf development. To be specific, it increases the serration of the leaf and create a more elongated leaf, and suggests that NATM has a function as a role in shaping leaf morphology. However, the NATM loss of function phenotype is not yet known. As mentioned before, Arabidopsis thaliana is a model plant that is used to study NATM expression. It has a simple leaf. As you can see in the figure here, it has a simple and undivided leaf, while Cardamine hirsuta is also a model plant that is closely related to Arabidopsis thaliana. However, it has a compound leaf with distinct leaflets. This difference in morphology between the Arabidopsis and Cardamine offered the opportunity to study the role of NATM in different species and different leaf morphology, and can increase our knowledge in genetic mechanisms of leaf shape and biodiversity. So, the aims of this study is to generate a CRISPR-induced null mutation in Cardamine hirsuta NATM using CRISPR-Cas9 gene editing. The second one is to examine the loss of function NATM mutation from previous project, and the last one is to observe and compare the function of NATM in both plants. And so the hypothesis of this study is that NATM function is conserved across different plant species. And if it's conserved, then the inactivation of NATM in Arabidopsis thaliana will result in the reduction of leaf striation, while the inactivation of NATM in Cardamine hirsuta will result in the reduction of the complexity of the compound leaf. To reach the aim of generating a CRISPR mutation of NATM, there are several steps. The first one is to design the gRNA of the CRISPR-Cas9. To do this, the genomic sequence of the cardamine is obtained from the Cardamine hirsuta genetic and genomics resources, while the Arabidopsis genomic sequence are obtained from TAIR. And the SnapGene software is used for annotation, while CRISPR is used to choose a suitable gRNA. The next step is transformation. To do this, 60 seeds of Cardamine hirsuta will be grown until flowering, and the transformation with Agrobacterium will be done using the floral dip methods. And then the successfully transformed T1 Cardamine hirsuta will have a fluorescent seeds, and the screening will be done using a fluorescence stereo microscope. Next, the screening of CRISPR-induced mutation or genotyping will be done using PCR-based RFLP. The first step is PCR using the NATM primer, followed by digestion using a restriction enzyme. The restriction enzyme has a recognition site inside the CRISPR target. And if the plant is wild type, then there will be no changes inside the CRISPR target and the enzyme will cut. However, if the plant is mutant, there are going to be changes in the CRISPR target and the recognition site will be altered and therefore the enzyme won't cut. You can see the difference with different pattern when visualized in gel electrophoresis. The next part is the observation and comparison of NATM function or phenotyping. To do the Cardamine hirsuta phenotyping, the leaflet number of the first 7 rosette leaf will be counted and compared. Next, the 5th rosette leaf of each plant will be scanned and the mean leaf length and area will be measured using the ImageJ software. The phenotype changes of wild type and mutant plants will be observed and compared. And statistical analysis of one-way ANOVA will be done using the CraftPath software. And for the sample size, it is 10 plants for each wild type and the NATM mutant. And the phenotype data for Arabidopsis thaliana will be obtained from previous study. For the feasibility, the Burn Lab has experience with CRISPR mutagenesis in Cardamine hirsuta. It is also well equipped with molecular biology lab and plant growth room. There are, however, a potential challenge of low transformation efficiency of CRISPR for NATM in Cardamine hirsuta. And lastly, for the expected outcome, the result of this project is expected to increase our understanding of NATM's role in leaf morphology of simple and compound leaf, and our understanding in NATM's expression across different plant species. And this understanding of NATM function in both species can have a great application in agriculture. For example, for the modification of leaf shape that can improve the photosynthesis efficiency and optimize the leaf arrangement in the canopy and resulted in increased crop productivity. This is the timeline of the project with the indicator of completion. And this is the reference that I use. Thank you for listening.